Mussels people are interested in, obviously, mainly as a food. But if you buy fresh mussels, you'll see this little bit of hair that is sticking off of your mussel. And actually, this is the byssus. People know it as the beard. The byssus is kind of the literal lifeline of the mussels. And the mussel has evolved these fibers as a way of really sticking very strongly in this environment where crashing waves are constantly coming and trying to rip them off these surfaces. Even though it has this incredibly strong attachment, the really wild thing we realized is that some mussels would actually jettison their entire byssus. And the mussel can do this essentially instantaneously. So these two things for us just didn't match. What makes this so strong yet easily releasable? My name is Matthew Harrington. I'm an associate professor at McGill University in the chemistry department. Our group is called Bio-Inspired Processing. We try to understand how living organisms make materials and how we can learn from them to make our own materials better. I've been studying mussels since 2003. So because we have these 20 years of experience studying this system, we can start to ask really deep questions about how this all works and how this is made. The byssus is not a living tissue. It is made up entirely of proteins. The fibers of the byssus are incredibly tough. They're as tough as Kevlar, and they're extensible. They're stretchy. They can stretch to double their length. So all the individual fibers are attached to the stem like branches on a tree. And then the stem is anchored into the living tissue like roots of a tree. The connection between the living muscle tissue and the non-living byssus can be very strong when it needs to, but it can also be released with apparently no force. What the mussel does is actually let go of the roots so they get pulled right out of the living tissue. Understanding how nature has successfully designed such an interface has a lot of benefit for inspiring engineers and material scientists. And so we decided to get a closer look at these tissues. We really quickly saw that the stem divides into about 40 to 50 different sheets. We call these the lamellae. We could see the living tissue, the non-living lamella, and then something in between them that we didn't know what it was. Once I saw that, I was really intrigued, and I felt like we could do something really interesting with this technique called focused ion beam scanning electron microscopy. You can reconstruct these images into 3D models that allow us to see these tiny little features. In the living tissue, you have extracellular matrix. And what's between the lamellae and the extracellular matrix are cilia. So cilia are like tiny hairs that are on the surface of living cells. There are literally billions of these tiny little hairs just in perfect contact with this non-living tissue. So it was really clear to us at that point that there was something going on with the cilia. We think that the cilia are only having very weak physical interaction with the lamellae, but it's the fact that there's so many of them. There's billions and billions of these interactions that then create friction that allows it to be so strong. It's analogous a little bit to gecko adhesion. So gecko feet are covered with these tiny hairs called setae, and they can use this to scale walls and crawl across ceilings. And by combining the strength of all these hairs, they get friction. So actually, it's not that surprising that the interface between the cilia and the lamellae is strong. What is surprising is that it can also be released with apparently no force. When we started noticing the role of cilia in this whole process, we found this paper that showed that cilia on the gills of the mussel use neurotransmitters to increase the speed of beating of the cilia. So we injected the serotonin, and then the thing just popped right out. And I was astonished, honestly. We were able to see that if we added more serotonin, the forces that were required to pull out the stem got weaker and weaker. We actually dissected out the tissue. The cilia were beating, and then when we added serotonin, they started beating faster. When we added metergoline, which was a serotonin inhibitor, they slowed down. So it really convinced us that these cilia are actually the critical players in this whole process. Maybe the sensing of this cue, whether it's chemical, mechanical, temperature, or whatever, can then trigger the release of a neurotransmitter that can then very quickly execute this plan of let's let the byssus go and allow the mussel get out of danger quickly as a mussel can get out of danger. You know, it's really seldom in a scientific career where the pieces come together so nicely, but I do see that down the line there might be some potential impact on the way that humans design implants or other advanced biomedical devices. It's just an incredible system that has been evolved through nature. And you know, every time I think we've got them figured out, every time I think now we know everything, there's a new curveball gets thrown at us.